The process of protein synthesis is made of two steps: transcription and translation, and they happen after each other. So we start with transcription. During transcription, the first step is that we have a messenger RNA copy of the gene that is made. This messenger RNA now carries the encoded information and leaves the nucleus into the cytoplasm. How the RNA is made: remember, first the DNA is sense strand and antisense strand. The sense strand runs from 5' to 3', and the antisense strand is going to run in the opposite direction, 3' to 5'. So we have transcription of the DNA into mRNA inside of the nucleus. And what we transcribe is actually the antisense strand. Every gene has 3 regions, and they occur in the following order. The first region is the promoter. We have an enzyme called RNA polymerase that comes and unwinds the 2 strands of the double helix. This is the promoter region. The second region is the coding region. It is right next to the promoter. This coding region is what contains the actual information for synthesizing the protein. Nucleotides of RNA are going to come and synthesize themselves on top of the coding region of the antisense strand, and they're going to be complementary to this antisense strand. So, one of the differences between DNA and RNA is that RNA is made of single short nucleotide strands, while the DNA is made of 2 nucleotide strands. The sugar in DNA is deoxyribose, while the sugar in RNA is ribose. The D in DNA stands for deoxyribose, and the R in RNA stands for ribose. Another difference is that the nitrogenous bases of DNA are adenine, guanine, cytosine, and thymine, while in RNA they are adenine, guanine, cytosine, and uracil. So thymine in DNA and uracil in RNA. So free nucleotides are going to assemble complementary to the antisense strand in the DNA in the coding region, and the base pairing rule as we know it: A is complementary to U. Remember, we have uracil and not thymine in RNA, and G is complementary to C. So in DNA, we have A complementary to T and C complementary to G. In RNA, A is complementary to U and C is complementary to G. And now finally, we have the third region, which is the termination sequence. That's where the synthesis of RNA stops. Now we have this mRNA. It is complementary to the antisense strand of DNA, so it is exactly like the sense strand, except for the fact that it has uracil instead of thymine. So this was the process of transcription. We were synthesizing mRNA complementary to the coding region on the antisense strand. And now this mRNA can leave the nucleus to go into the cytoplasm, to go into the next step, which is translation, where it will make proteins. This is the messenger RNA, and this is transcription. Here it is leaving the nucleus into the cytoplasm.